I think it's time to continue this deep dive in unsupervised learning. So this afternoon will be all about Ylenia. So she will give you both the lecture now and then also the hands-on session after the coffee break. Okay? So let's resume. The floor is yours.
Thank you very much. So, welcome. Also, sempre difficile. It's very difficult to start again after lunch or after the breakfast in the morning. Okay, so welcome to this part of the last part of the Unsupervised Learning session. As this morning I was telling you, we will speak about other clustering algorithms, and then we have the final session that is an hands-on session in which you have to try to solve some problems on some very simple, not very simple data set that I generated. So here we left off this morning. We were saying that the k-means is very able to, yeah?
Someone online who has asked, so we have seen a hands up online, so if you have something to say, please speak. No? Ok. Ok, no, sarà, sì,
Sbaglio. No, don't worry. So we were saying that this morning we saw the k-means that is very able to find clusters that are in a circular shape, for example, but it's very difficult to work with this kind of data set. And as we have seen in the notebook at the end of the first session. And so how can we solve this problem? Well, we have to change the algorithm and use another algorithm. For example, this the DBSCAN, that is a density-based clustering algorithm, which is based on density. The name says density-based spatial clustering of application with noise. So this DBSCAN help us to go beyond the problem of the circular shape, and it works on the density and on the idea that points that are close together in the high density region are part of the same cluster, but this cluster should be also separated by lower density regions, okay? So this is the idea of the DBSCAN, and it differs radically from the k-means because it doesn't look for the centroids, so we don't have the problem of searching for centroids of the clusters, because he works on the density. And so, for example, and so we need other parameters. We don't need the number of the clusters a priori, but we need different parameters. In particular, the DBSCAN introducing the concept of the radius epsilon, and it is based on the distance, so on another metric, on the metric of the distance. In fact, given a distance metric d, the neighborhood of a point P is defined in this way. So we have this set of data that are all the points that are inside within the radius epsilon. And then how dense could be a cluster, a group to be considered as a cluster. The second notion is the density. Is translated by the mean points, mean PTS, that indicates the minimum number of observations that are required for a neighborhood of radius epsilon to represent a sufficiently populated region. So if we have, if we tell to the algorithm, we need to cluster all the points, we need to consider it as a cluster, a group that has, for example, five points, at least 5 points within a radius 0.1, then the DBSCAN will group this data basing on these two parameters. Okay, so the idea is very simple. But before to understand how mathematically the DBSCAN works, we have also to introduce 3 other concepts: the core point, the border point, and the noise point, because the difference with the k-means is that not all the points are considered within a cluster. So we can have the noise points that are outside the cluster. So they are points that are not considered within clusters, okay, within the groups. So the core point is that the point that contains at least mean points, the number of mean points within the radius. And so the algorithm starts from this core point to expand the cluster, and the border point is a point that has fewer mean points around it, around in its neighborhood, but is within the epsilon, within the radius that we defined before. Okay, so these points usually defines the edge of the clusters. And then we have the noise point, that is the outlier, a point that is neither a core point nor a border point, and these are automatically ignored by the clustering processes. Okay, so these are the outliers, and tomorrow you will see also that the DBSCAN is one of the algorithms that is very useful that is adopted to do anomaly detection. So let's start from this data set. We have a dummy data set. We have 2 regions here and a point in the center. How do we expect the clustering, the cluster works? Well, probably we have 2 clusters, cluster 1 and cluster 2, and the point at the center should be the noise point. Okay, if this data set if we give this data set to the k-means, I don't know if it is able to cluster in this way the points. We can try. And so the goal is to cluster this point is to densely packed clusters. So that remember the density. We should work on the density. So let's start from the beginning. We selected a random point P, and then we draw a radius, its neighborhood with the radius epsilon that we give at the beginning of our algorithm, and then he counts how many points fall inside the circle. Okay, and so these are the yellow points around the green one, and then he counts. He asks himself, the points within the radius are major or not the mean points that I'm giving to the me, data scientists, are giving to the algorithm. And then he colored all the data. Okay, these are points within the radius. They are major then. So this could be a first cluster. But he goes on and check the other points in the cluster in order to see if there are in the same neighborhood other points to be considered in the same clusters, and so and so. If a point is itself a core point, Then all the neighbors, sorry, are included in the current clusters, okay? Then there is, for example, the algorithm, for example, finds a point that is a border point because, as you can see, there are not some many points within its radius, and then this is a border point. And so the border point is included in the cluster, but the clustering stops here. Checks if it can stop, and then it goes on with all the points, okay? So this process is goes on until all the points are part of a cluster. And then it starts with the other side, with the other points of the other side in this case. And then at the end, he finds the unique point in the center of this image. And this is neither a core point nor a board point, so it is a noise point. So this is how the DBSCAN works, okay? And at the end, we have that all the points available, that is -1 if it is a noise point. Otherwise, we have 0 or 1 or 2, in this case 0 or 1 if the point belongs to the cluster 0 or the cluster 1. So how to choose epsilon and mean points? Because with the k-means we have the problem to understand how many clusters do we have at the beginning. But now with the HDBSCAN we have to understand from our dataset what is the best choice to do for with the epsilon and the minimum number of points necessary for considering a group as a cluster, a group of data as a cluster? Because the problem is that if we choose too small epsilon, then we tend to classify many points as outliers, because we can consider the clusters that are too small, and so the density, we are selecting a density too concentrated in some parts of the dataset, and so many points could be considered as noise. Otherwise, if we consider too large values of epsilon, then we can merge distinct clusters. So we can if some clusters are near in the space parameters, then, for example, they become only one. They are not. It is impossible to distinguish them. And at the same time, also the mean points is very important, because if the mean points is too low, then it is too permissive, and so we have spurious clusters. Otherwise, we have the method that is too conservative, so we have more noise and more difficult clusters to be formed. And so also in this case, we have to adopt the matrix, the silhouette score, for example, or the k-distance plot, as in this case. The k-distance plot adopt the inertia that it is similar to the elbow method that we have seen this morning for the k-means, because we have the distance on the y-axis, that is the distance of each point to its nearest neighborhood within each within the cluster. So we fix the k that is equal usually to mean points, and then we sort the distances that we obtained between these data, and then we see where there is the elbow of points at the end. And this is important because, as you can see here in these two figures, you can see that also some very small variation of this epsilon parameter can give us very different results at the end, okay? with an epsilon parameter that is small, that is 0.12 and not 0.14, you can see that we have the fragmentation of the clusters, and at the end we have this problem, because visually we can see that there are only 2 clusters in this plot. And this is a problem that I reproduced in the notebook that we will see after the coffee break, because I prepared a dataset in which we have the 2 distribution and then a cluster that is very near, and at the end it is very easy to fragmentate the clusters and to not have the final solution that I myself implemented in the algorithm. So these are the problems with the DBSCAN. However, the advantages are that we can discover arbitrary morphologies, and we have seen that we are working with a very particular shape of clusters, and this is different from the k-means. But for example, we have seen this also in the notebook, in which we have these 2 shapes that are very particular of clusters, and how they k-mean clusters the data, and how the DBSCAN at the end clusters. But here you have to note that there is a very the density of the data is quite well distributed. We don't have a variation of density, okay, within the data, and this is also important. We don't have to specify the number of clusters, but we need to specify the epsilon and mean points at the beginning. So, but this is an advantage. And also, it is very robust for finding the noise and outliers. In fact, as I anticipated you, it is one of the algorithms that is very used, very adopted to work for anomaly detection, also in our field of application. Because all the points, not all the points are inserted into the clusters, and so this is the difference with the k-means. And finally, also in this case, there is a great interpretability of the parameters, because we understand well what is an epsilon, the radius, and the minimum number of points that are necessary to understand if a group could be considered or not a cluster. And the DBSCAN is very used for the geographical clustering. For example, if you have a data set in which we have the longitude and latitude coordinates, you can do a cluster, and the DBSCAN works very well because it is based on the Euclidean distance, but you can implement also other metrics, and then you can cluster the points on the geographical with the geographical insight, and map them on the geographic maps, and then understand if there are points that are very interesting for doing, I don't know, the DBSCAN is usually adopted also by Google Maps and so on. At the beginning, I know that there was an algorithm such that, in order to understand if these groups, if this data, to understand it also for the traffic, or to find particular point of interest for tourism and so on. Then the disadvantages are that is high sensitive to the parameterization, as we have seen the epsilon that change by 0.02 and give us different results. And also it is very sensitive also to the changing in density. And this part is well understood when we will see the hierarchical DBSCAN that try to go to solve this problem, because in this case we are working with density that is quite uniform. But if we have a not uniform distribution of points within our parameter space, this could be a problem, because the DBSCAN finds the minimum points within a radius, but then if there is some distances between one point and another one, this could be considered as a new cluster, and so it doesn't belong to the cluster, maybe, and maybe it is only a sort of projection into the parameter space. The non-deterministic boundary assignment. So while the identification of the core points and noise is deterministic, the assignment of the border points may depend on the processing order of the data. And so, for example, it is if a point is reachable for multiple clusters, it could be a problem. And the curse of dimensionality, because this could be a problem in high dimensional spaces, where the distance between points become less meaningful. And so in this case, the density-based metrics lose their discriminative power unless we perform the dimensionality reduction, okay? And so sometimes we need to perform before the dimensionality reduction. So how to solve this problem? Because this is a kind of solution to solve the problem of the k-means, but we can have also other way to cluster the data. And for example, the HDBSCAN is one solution, but before to understand the HDBSCAN, that is the hierarchical version of the DBSCAN, we need to introduce the hierarchical clustering. That is another way to introduce to group data, but it is based on a different way to organize the data, because at the end we have the organization of the data that changes. And so we are going from density that is used from by the DBSCAN to hierarchy, in which we have the data that are organized in nested architectures. For example, we can think to some phenomena that have this kind of structures within data, and so we because, for example, there are difference in the density distribution, we have subgroups within the data, and so on. And so in this kind of problems, we need to treat the data at different level of granularity. We have to understand how the data is distributed at a granular level. And for this case, we can use the hierarchical clustering, okay? Because we have a level representation of our data set, and it is done through the dendrogram. But let's start from the beginning. The hierarchical clustering is always unsupervised, and it doesn't want to have a fixed number of clusters at the end, but he tries to build a cluster hierarchy within our data that is a tree-like structure. So I think that we already know the dendrogram. The dendrogram, that is these tree-like structures. And the idea is to understand how the points progressively aggregate. So if we start from the bottom part, so where we have the data points, we can start from here and to understand how the points cluster progressively going from the bottom to the top, or starting from the top, so starting from the clusters, how they divide into the single data. In fact, the hierarchical clustering is, it is because I set it in the how the points or clusters progressively aggregate or disaggregate. Because we have two kinds of hierarchical clustering, but we have to just a few notions. Each leaf represents a point at the end, so these are called leaf. The internal nodes represent the mergers or the split, if we are going from top to bottom, and then the height of the branches indicate the distance between the merged clusters. So it is in scale. Okay, so we are using the distance, and it is on the y-axis. At the end, we need to understand where to cut the hierarchy to obtain the desired clusters, because if, as in this case, set the line at 4 point some and 3 point something, then we have only 3 clusters selected within our model. But if we set a threshold that is at 2 point something, then we have 1, 2, 3, 4, 5 clusters selected. So the point here is to understand where to cut the hierarchy in order to obtain the desired clusters. How to do that? Always we have to inspect the data, and the data scientist is always the person who can solve this problem, because we don't have to only to perform machine learning in general on a data set, but we need to understand what the algorithm is doing and the output that we have that we are obtaining. And so we have to go and see if these clusters are in some way good for our scientific purposes, because we need always to, before doing machine learning, we have to make us a question: what I want to solve, what I decide, what I want to obtain from this experiment, and then I have to understand if the data are telling me something that is good or not, but in a scientific also way. Okay, so here we have also to understand how where to cut the hierarchy to obtain the clusters. And so when as I was saying you, there are two kind of hierarchical clusterings that are these are two strategies: the agglomerative clustering that comes that starts at the bottom, so each point is each leaf, each point is a cluster, and then he goes iteratively, he tries to group similar data within the clusters, and then it calculates the distance, and he groups the similar clusters iteratively, and at the end we have only one clusters, the unique clusters in which we have all the data grouped. And this is the bottom-up strategy, the agglomerative clustering. And then we have the divisive clustering, which we start from a single cluster, that is the whole dataset from which we are starting. And then at each iteration, the algorithm tries to divide a cluster into subclusters, and then it goes on until we have too many small clusters or single points at the end. Okay, and so in this way, also in this case, this hierarchical clustering is trying to measure the similarity between points or clusters, and so there are also some metrics that we have to define in order to understand how these points could be similar or not. The most used is the agglomerative clusters. In fact, I want to explain you in detail how it works. And just a very simple example in order to understand what are we dealing about. Because, for example, we have here different kind of animals: the eagle, peacock, lion, bear, spider, and scorpion. And how to the hierarchical clustering works. The agglomerative starts from each animal at the end, and then, okay, I classified the first two in birds, then mammals, then more than 3 legs, spider and the scorpion. So we have 3 clusters here. Then we want to agglomerate also these clusters if it is possible. Birds and mammals are vertebrate, the other ones not. And it remains without, not vertebrate. We have a class that is a group that is vertebrate. On the other hand, no, they are only animals with more than 3 legs. And then animals at the beginning, because it is the final cluster, okay? But at the same time, we can do it by starting from the animals at the top of the figure, and then do the inverse process in order to find the unique animals at the end. So this is how the hierarchical clustering works. In a mathematical way, that is a little bit difficult. We have the 5 points that are separated and are 5 clusters. Then we have to calculate the distance between clusters, and so that initially are individual points. So we need to calculate the distances between the points, and then we merge the clusters that have the smallest distance. The pair of clusters with the smallest distance is merged according to the linkage rule. So the linkage rule, I will show you this linkage rule because these are the metrics that will be used, and they are very, very important. And then we updated the distances. So we updated the distance matrix, calculated the new distance between the new clusters and all the other one. And then we repeat it until the process ends, and until the points are in a single cluster, okay? So this is how this agglomerative clustering works. And what are these linkage measures? Well, we have to measure the distance between the clusters. But how we can do it in the hierarchical clustering? We have to choose these metrics, and there are different linkage measures. The single linkage measure help us to calculate the distance between the minimum distance between the points that are in 2 different clusters, but we have to find the minimum distances between these clusters. So it is the distance between the closest pairs of points belonging to different clusters. And it is clear that it helps to, it favors the elongated chains and clusters because just a pair of nearby points is enough to emerge 2 groups, no? So this is the single linkage. The other linkage is the complete linkage that uses the distance between the farthest pair of points, always belonging to different clusters, and it will produce the more compact clusters, okay? Finally, we have the average linkage, that is the calculation performing by average all the distances between the points in the first cluster and all the points between the points in the second cluster, okay? And so usually it is the matrix that is most adopted when we try to build a hierarchical clustering. Because it is a compromise between having a compact clusters or not. We have to go one step back, but only to understand the kind of data that we are using and what we are trying to solve. Because with the DBSCAN, we see that we are working on data, that the DBSCAN works well when the density are uniform within the dataset. But in this case, for example, you can see that the dataset has not a uniform density, and here there are the main difficulties, because how should we choose the right epsilon to understand where the cluster ends and the mean points, to how to understand if a point here belongs to this cluster, or here there is another cluster or not. So the DBSCAN not always is not able to understand how to manage these kind of clusters. And so here comes the solution, that is the HDBSCAN, the hierarchical DBSCAN, that what is done, what the hierarchical density-based spatial clustering of application with noise, what this algorithm do is to combine the density with the hierarchical construct to select the stable clusters, even when the density varies. In fact, from these structures, we can see what are the clusters are 5, and also the HDBSCAN tries to select these clusters basing on how much they are stable. So there is another notion that we are introducing, okay? So the k-means works on the shape, with the circular shape, and they are centroid based, and we have seen how. The DBSCAN is working on the density, and it has to use the epsilon and the minimum number of points. Here we are introducing another step, that is the density with also the hierarchy, the hierarchical construct, and the notion of the stable, of the persistence of the clusters, how persistent will be these clusters, and how to translate it into mathematical way. Well, the key parameters of the HDBSCAN are the minimum cluster size, so we have the minimum threshold of points that are required to form clusters that are similar to the mean points of the DBSCAN, but also the mean samples, and it helps us to define how dense a point must be to avoid being considered as an outlier. So this is the change of the perspective here. And some useful outputs of the DBSCAN could be the labels. -1 is noise. The other are the labels of the values, and the probabilities. The probabilities of the cluster persistence, but there are two versions of HDBSCAN. One of scikit-learn that we will use, but there is another HDBSCAN that you can import. You have to install as another packages in your environment that can give also the cluster persistence. The probabilities is the strength with which each sample is a member of its assigned clusters, okay? So how to measure how good is our clustering? We have to analyze the probabilities of each point to belong to that clusters. If the probability is high, then it is a good point for that clusters, otherwise not. The cluster persistence is a score of how persistent each cluster is. And the persistence, I will explain you in very detail this concept of persistence. I have a few slides because I want you to understand well how this HDBSCAN works, because it is a very good algorithm to be used for clustering and to perform outlier detection. So let's start here, from here. 57, ci abbiamo ancora, sì, ok. Ok, because I want also to show you in detail the code, because then you can work on it. So let's start from this data set. How many clusters do we see? I don't know if I choose 6. Okay, see? Yeah. There are 6, it is easy to distinguish 6 clusters. And how we can define these clusters? We can define it by looking at the density of the data. They are too concentrated in some space. So there are highly dense regions that are separated by sparse or noisy space. Okay, so this is the definition of the clusters. Okay, example number 1. We have a 1D simulated data. So we have the strip plot of X on the top of the slide, and here we can see that there are the distribution of the data is in part dense. There are 3 dense regions and 2 sparse regions, and in this case we can separate 3 different clusters. Okay, it is the definition that we have just given in the first in the previous slide. So in this case, if we look at the PDF, the probability distribution function of the same variable, we can see that we have 3 peaks. And then we have the sparse regions that are those ones at the bottom of the distribution, right? Okay, example number 2. The strip plot of X is at the top, and how many clusters do we see if you don't look at the probability distribution here at the bottom? If we look only at the top, our eyes see only 2 clusters, because it's very difficult to separate the 2 clusters on the left, right? But if we see the probability distribution function, well, here you can see that there are 3 peaks. But how to decide where to cut? And so how many clusters do we have? This is the problem of the hierarchical clustering, okay? Not because it is very difficult to understand where to cut. But if we do this kind of exercise and we define a threshold here, we can see that, well, there are, if we use a threshold that is very low, we have only 2 clusters, otherwise we have 3 peaks. But if we, if the HDBSCAN should work on this kind, in this way, we are only giving another parameters to the algorithm, and so we are translating the problem, because we are going from the decision of the epsilon and mean points to, we are all adding another parameter that is the threshold to which the algorithm should work, okay? And the idea is to consider the area, the sum of the areas, and in this case, as you can see, we have in these two different examples the area that are the sum of the colors, okay? So you have to consider the colors. If blue plus, in the first example, if the blue and yellow area are majors than green, as in this case, then we have two separate clusters. So otherwise, here we have an area. Grazie. Okay, so the blue and the yellow one are less than the green area, and so the area of the green is much larger, and this means that they are just bumps, so they are not clusters. The area of the regions is the measure of persistence. So how much a cluster is persistent mean how much the area are greater or not than the sum of the green parts. So where we are cutting the shape of the distribution, or the probability distribution, okay? And this method is the excess of mass, in which we have to maximize the total sum of persistence of the clusters under the constraint that the chosen clusters are not overlapping, because we have to pay attention to the peaks. We have to not overlap the two, some Gaussians, okay, the two curves of the two peaks. And then after that, so we have to get, we are getting multiple levels at different values of lambda, of the parameter, the threshold. So we are not defined a lambda, but the HDBSCAN will work on different lambda. It will change this parameter, and he have to understand where to separate or to aggregate these clusters, these peaks, and then he has to translate it into a dendrogram, that is the hierarchical tree representation that usually is done from, it is represented from, in which, in trees, in which we have the greater level at the top, and then the bottom one with the, okay, it is the tree representation on the, from top to down. And I wrote here the idea is that when we have a sea, and then the level of sea is going down, and then you can see all the islands that are coming up from the sea, and then we have, and it's the same similitude to understand where to cut how the HDBSCAN is working in order to understand where to cut the groups. Well, how to translate it into mathematics? We need to introduce two concepts, the core distance and the mutual reachability distance, okay? So the core distance of a point is defined as the Euclidean distance to its k nearest neighbor, and where k is governed obviously by the minimum samples parameters. And so, the concept of the lambda, the values that we have to, where to cut the threshold, we have to, where to, the HDBSCAN have to cut the clusters, is defined as the inverse of the mutual reachability distance, that is defined this way, by given a pair of points, I wrote P and Q, but the formula is with A and B. I'm just seeing that. We have the maximum between the distances, the core points, the core distances of A and B, and the distances between A and B. But we have to understand that this mutual reachability distance help us to work as a metric repulsion mechanism, because it is defined to in order to measure where to work as the Euclidean distance when the 2 points are near, because we are in a high density local context. But when we are in sparse regions, the distance undergoes a constraint deletion of this point, okay? So this mutual reachability distance help us to have the help us to have the segregation between the points that are outliers. Otherwise, they are considered as nearest points, and so they are considered as points of the same cluster. So it helps us to preserve the topology of our space parameter, and this is very, very important. So how it works? First of all, we have to define the minimum cluster size and the minimum sample. The minimum cluster size is written in this way, MCL, in the text. And then to calculate, we have to calculate the core distance and then the mutual reachability distance of each point. Then we have to build the minimum spanning tree. That is the graph that we built using the distances of the using the mutual reachability distance. How? We have to connect all the points on the left. We have this distribution of the data, and so we have to connect all the points, and we don't use the standard Euclidean distance, but the mutual reachability distance. And how to do it? Because we connect 2 points A and B, and we weight them for their mutual reachability distance, and this help us to push the way the outliers that are those points that are too at the periphery of the cluster, so they are too distant one from another one, okay? So this is how to build the minimum spanning tree. After that, we convert the minimum spanning tree into a hierarchy, that is the dendrogram of the connected components, and this is how the HDBSCAN do it, because the minimum spanning tree is the precursor of the dendrogram. It sorts all the edge between within the minimum spanning tree, and they are sorted in an ascending order of weight, okay? And then we try to construct the dendrogram is constructed from the bottom to the top of the figure, as you can see from this graph, and we merge all the points initially into microclusters, and then we go further in order to build the clusters with we aggregate the clusters using the notion that we introduced with the hierarchical agglomerative clustering. Okay, so here is how the HDBSCAN is working, basing on the hierarchical idea, and not only on the density. After that, after we have the dendrogram, we have to build the tree condensation, because the branches that contains fewer points, then we have to decide if they are noise or not. And so if these branches have fewer points than the minimum cluster size, then we have to falling out. The HDBSCAN treat them as noise. And so here we have the identification of outliers. And in this way, we are cutting the branches. And so we are simplifying the hierarchical structures. And okay, so I think that I'm saying you these things that I wrote in this slide, because yes, if a branch contains fewer than the minimum cluster size of points, then these points are fallen out of the parent clusters. And if both branches contains at least the minimum cluster size points, then the split is considered legitimate, and so we are there is the birth of two distinct child clusters. Okay, and finally, and this is how we have written what I have written here. Finally, how to measure the famous cluster persistence, because you remember the area that we selected, that we colored in the map in some few slides before. And the excess of mass help us to calculate the area of these branches when we have built them within this graph. And as you can see, there is the base, that is the width, that represents the number of points that belongs to the same cluster. So this is the base. And the height, that is lambda, that represents the persistence. So how persistent, the first question I do before was, how persistent must be a group to be considered as a clusters? And so this is given by the height, the height of these groups, that is given by the inverse of the distance of the mutual reachability distance. Okay, and so a cluster is considered stable if it manages to maintain a large population with and a significant density range, that is the height. So this is how it decide if a group is a cluster or not. And so you can see here that here this cluster has a high stability, the one of on the left. This one have a medium stability, these selected clusters, while these clusters is a discarded one because it has a low stability, because the area is not so high basing on the other area. Okay. So we have at the end to combine all these results, and at the end we have the calculation of the clustering of our clusters. We have at the end the results of our clustering. As I was saying you before, we have two ways to perform the HDBSCAN. The HDBSCAN that came from the scikit-learn doesn't have the possibility to visualize, for example, this kind of details, because they haven't yet implemented the condensed tree, the dendrogram, and so on, and so we can't visualize them. But if you use the HDBSCAN, if you search on the internet HDBSCAN, it can be seen how to import, to install these packages, this package, and then to import it. It is very simple. If you want, we can do it in the afternoon, and we can see also step by step how the HDBSCAN works, and it could be good to see all this graph, because you can also understand how your data have been clustered, okay? Sì.
No, it's just for the people online.
Oh, thank you. Sorry for previous page. How did you found that which one has the lowest stability and which one has the highest stability? I didn't understand this part. Sorry. If you can,
This one,
yes.
the stability, because the stability is defined in this way. So you have to see the area of the groups, the base that represents the number of points, and the height that represents the persistence. And finally, the stability of each cluster is defined as the sum of all the stability of the children. And then if you see that this area is, for example, this area is less than the sum of the children, and then it has a lower stability, and so it is discarded as clusters. And the other ones have are not, because they have high stability. Okay, so we have to maximize the global stability at the end.
Okay, thank you.
You're welcome. So the advantages are that we can detect different clusters, and we can detect these different clusters with different variable density, as in this case, and we have also the identification of noise between the clusters. Then another advantage is the fact that we are reducing the arbitrary parameterization, because we have to not work on a threshold, but we have to use the mean cluster size and also the mean points. But the mean cluster size is that is the parameter that it is the most important parameter we have to choose within the implementation of this algorithm. And also there is an advanced noise filtering, because using the mutual reachability distance makes the HDBSCAN a very robust algorithm, okay? And so it helps us to understand if there are some points in low density areas that can be mathematically pushed away from clusters, and the HDBSCAN help us to understand the reality of these, the fact that these points are really outliers, are noise, and not they do not belong to any groups, any clusters. And then we can also see the hierarchy. We can see how our dataset is clustered step by step, in order to see, to understand also how this data is composed, and it can be useful, because we can see at different levels how the clusters emerge or dissolves. within our data. So it is very, I think that it is very useful to see this kind of representation of our data set. And then the stability, in this way, thanks to the persistence that we have introduced in this algorithm, we have the we know that the clusters, if some groups are selected as clusters, is that because they are persistent and because they survive to the longest during the condensation process. So this give to the HDBSCAN a very important aspect, and it is a good candidate for to be used in for different kind of experiments. Another this is a disadvantage, the fact that is computational and the computational memory costs. But remembers that the HDBSCAN, the difference with the DBSCAN is that the HDBSCAN could be parallelized, so we can use it. We can split the processes, okay, while the DBSCAN is not parallelized, and this is a problem because it has a very computational cost. But here we need these processes could use a very high processing time, and also the RAM usage can become critical. It is very sensitive to the dimensionality, to the curse of the dimensionality. In space, for example, that has hundreds of features. So in high dimensional space, this could be a problem. And the fact that is dependent from the distance matrix. So when we are given a data set, and we have to choose the right distance metric, in order to understand how similar these points are, because you have to remember that the distance matrix, the similarity matrix that we used at the beginning, is shaping our parameter space at the beginning of the machine learning algorithm. And it doesn't depend on the kind of what kind of algorithm we are using. Okay, it's independent this fact. This choice is very important. And then there is the difficulty with overlapping clusters, and I choose an algorithm, an example. There is an exercise in which I choose I implemented a data set in which we have two different forms, and then a cluster that is very near to another one. And so here there is the problem to separate these clusters, and you can see how the real life is. Okay, I don't know. Okay, 10 minutes, so we can start. Okay, so if there are questions, we can answer you. Otherwise, I can start to see the notebook.
Thank you very much. Just simple question. I didn't understand what you said. You can choose to do a bottom-up, top-down approach. I didn't understand if you obtain the same results based on the pruning you were talking about, and is a choice a user make, and you obtain different tree at the end, or you always get the same tree either way you go?
No, sorry.
You mentioned you show bottom-up, top-down approach for building your graph,
okay.
right? So if you choose one or the other, do you get different results, or it's just an implementation question?
No, it's for the HDBSCAN or the hierarchical in general?
In general.
No, in general, no, it is not, but it is very useful to, I think that is, the agglomerative one is the most used and adopted in the community, in the scientific community. And I think that it is because we are starting from the data and then we try to agglomerate it, and it is more useful to agglomerate in this way.
You can interpret better.
Think, yeah, for the interpretation, I think also that it could be also. I don't know the mathematical implementation of the other one, if it is difficult and perhaps it has some computational costs that are greater than the agglomerative one, but I have to check it, and so I'm not sure of this answer, but it could be.
Questions?
Okay.
I want to ask about the dimensionality problem that this algorithm have. Is applicable to use an autoencoder to reduce the dimensionality and then apply the clustering in the latent space? Is something that is global, and or there is a theoretical problem to work in the latent space?
With machine learning, you can do everything you want. The autoencoder usually, for example, is used to reduce the dimensionality, and then you can apply, for example, a classification step if you know the labels, and so you can understand. For example, it is very used in the image processing. No, you can start from an autoencoder in order to extract the features in the latent space, and then the same feature of the latent space are then used to classify, for example, your data, but also with the cluster, because perhaps you are in an unsupervised way, so you can previously reduce the features and extract new features. And then to clusters, yes, you can do it. The only problem, but it is a problem only because you are starting from a feature space that you know. So it is a feature space, a physical feature space, I don't know, a statistical one, and then you are going to the feature space of the latent space. So you have some features that you are not, you don't know what kind of features they are, because they are not related to in some way to the physical feature space, and so it is a problem of interpretability of your of your problem. But at the end, you can have your clusters, and then you can decide if these clusters correspond or not to the problem that you are trying to solve, and then you can decide if this algorithm is working well or not. Yeah, you can do it. Okay. It's only a problem. There is only the problem of the interpretation of the features in the latent space, but it is not a problem, a real problem.
Okay, thank you.
You're welcome. So this is the notebook I prepared you. It is the Unsupervised Learning Part 3 that we can download from the last session of this day on Indigo. And here is what I want to show you, and then we have to work also on another notebook in which I prepared you some example. And so I generate the data set on which we have to work, but it is the same at the end of the other notebook. So the moon shape and the clusters on the bottom left of the graphs. And then we have to data process this data set. And then we have to understand how the DBSCAN and how the HDBSCAN works on this data set. And then we can compare also the results by inspecting them. So I import the needed packages, and they are the same of this morning, but here from the sklearn I'm importing the DBSCAN and the HDBSCAN. At the end of this notebook, I think that I inserted also the other library of the HDBSCAN. It will work in the same way, but it gave us the possibility to see step by step also the condensed tree and so on. And we should only pay attention to the input parameters that are set automatically at the beginning, because, for example, I don't know, but perhaps in the scikit-learn the first the Euclidean distance is that one used by default, but for example in the other library it is not the Euclidean distance, or the mean points by default is 0.01, and on the other library is 0.05. Okay, so in order to obtain the same results, we should pay attention to this change of initial parameters, only this thing. So I generated the data, and these are the irregular data that I generated. And as you can see, there is these two structures, like the moon shape, and then a clusters that is there. But remember that I told you in the last slides before that the clusters is is very near to a shape, and this could be a problem because we have seen that the HDBSCAN is not very able to separate this kind of clusters if there is okay, if they are very near. And so we have to pre-process the data. I'm scaling the data set, and so the visualization, it doesn't change the visualization, but only the scaling of the data set. So the shape is the same. And then we have to perform the DBSCAN. Okay, so we have to decide the epsilon, the APS is the epsilon radius, and how many points, the mean points, in this case, the name is mean samples. How many points do we have to consider, the DBSCAN should consider in order to label the point as belonging to the same clusters, okay? And so I choose these two parameters, the epsilon 0.3 and mean samples equal to 5. So a cluster should be a group of at least 5 points for the algorithm, okay? And so this is how we have to implement it, to implement the DBSCAN. Here is the simple way to create the model, the DBSCAN model, passing these two parameters, and then here we fit the scaled data that I called X new scaled, okay? And then here, at the end, when we run this code, we have the clusters, that is the clustering, we perform the clusters, and here at the end we have these two variables that gave us the number of clusters, how many clusters the DBSCAN has found, and the noise, how many outliers the DBSCAN is finding. And remember that the noise is, the labels of the noise for DBSCAN and also for the HDBSCAN is -1, okay? So -1 for the noise, otherwise we have 0, 1, 2, 3, it has the number of cluster, the label of the clusters to which each point belongs. And so I can also, here I said just to calculate the silhouette score. So how good is this clustering? And here I have a silhouette score that is 0.16, and if you remember, the silhouette score should be almost near to 1. So there is not, it is not so a very good value for these metrics. But when I plot our results with of this DBSCAN, you can see here that we are in fact that we are that the DBSCAN is selecting only 2 clusters, but they were 3 clusters, and so the value, the lower value of the silhouette score, okay? And then these are only 2 points of noise. I think that these results are not so good, okay? I don't know if you agree with me, but I think that the DBSCAN is not working so well. And so we can see in interactive way what is but I need to, sorry if it is not so good for the labels, but let me do this thing. Okay, in order to have all the plot in within the screen. Here we can see how our DBSCAN works when we change the epsilon and the minimum sample size, okay? If we try to, we have set the minimum sample size, okay, to 5. So I was saying to say the minimum sample size to 5, but if we try to insert 12 points, for example, then you can see how the DBSCAN is working. He's trying to separate the clusters on the top one, on the top of the moon shape, but it is not able to separate the clusters on this part of the plot. So how to do it? Well, I'm trying to represent 5, for example. I don't know what is the best solution in this moment because I don't remember. So I have to move, for example, the epsilon. And here I can see that there is a fragmentation and we are producing 12 clusters, for example, and 13 clusters, sorry, and the red points are in red. So as you can see, it is very difficult to understand what are the main, the best solution in order to find the optimal, the optimal clustering results, okay? We can try to see if it is works better in this way or trying to, but we are not very able to separate the 3 clusters that we have built at the beginning. And so this is the difficult part of our works in order to understand how to separate these clusters that we have built at the beginning of this lesson. So here is where we left off, and we don't know how many clusters the DBSCAN is able to select and how it is able to select also the clusters thanks to the epsilon, the mean points that are the two parameters. And in the following code, we can try to evaluate the metrics, the elbow, but we can see if the elbow will function or not. I think that perhaps I haven't applied the elbow because the elbow is usually is not useful with the DBSCAN because it evaluates the clusters based on the compactness, and this is not the case. And but we can use the silhouette score and the Davis-Baldwin index. In fact, in this part of the code, you can see how to do it, and if we execute, in fact, this part, we are trying to understand how what kind of values do we have to assign to the epsilon or to the mean.
Sorry, we are seeing the auditorium view, not the presenter view.
No, it is somebody who has the microphone open, okay? And so in this way, we can try to do this evaluation, and that at the end of this code, we can see that the best value of the best silhouette scores.
Scusate, non vediamo la presentazione.
The best silhouette scores are 0.3. The best silhouette score is 0.39 with these parameters, epsilon 0.4, and the mean sample is equal to 2. So the cluster needs at least 2 points to create a cluster. I have to, yeah. Okay. Yeah, it is a problem, the Zoom, when I have to show you the plot. So, okay, I can change this visualization every time. And the best Davis-Baldwin index is 0.37 with the parameters 0, 3, and mean sample. So, as you can see, the parameters could not be the same, and we have to see if these are good parameters and how the DBSCAN at the end clusters our data. So we rerun this cell in order to see the plot. So I organized the silhouette score at the variation of the mean samples and the epsilon. And as you can see, we have remember the silhouette score must be higher. And in this case, the maximum is the 0.36 that we have in the previous cell. And we can try to have these values with the epsilon around 0.5, 0.4, and the mean samples that varies between 2, 4, and 8, for example, or also here on the right. And otherwise, the Davis-Baldwin give the best values are the yellow one, because we have the Davis-Baldwin should be towards 0 to be good. And for example, we have 0.37 that is at 14 mean samples, and I think, yeah, and 0.3 epsilon, the epsilon values. So with this kind of parameters in mind, we can go into see if how the clustering works, and this is the final clustering. I think, sorry, this is the representation of the data. This is the final clustering when using the silhouette score, I think. Clustering with the best Davis-Baldwin, and this with the silhouette parameters. So with the silhouette parameters, we find only 2 clusters, and with the Davis-Baldwin, the final analysis give us the 5 clusters and all the red points that are considered as noise. So these are the best solutions for these for these two parameters when we consider this data set, okay? And here there is the problem in which we have to understand if our models is working well or not. Here there is the how many points of noise we have when considering the silhouette parameters. They are all there is no noise. There are only 2 clusters and there is no noise. And with the Davis-Baldwin parameters, the best results give us 87 outliers. So what is the best choice? We have to try to understand basing on our data, our features, and then we have to understand what kind of solution for us is the best solution, because these are metrics, but we have then to choose the right, what is the right representation of our data, basing on the problem that we have to analyze. The same thing on the same data set has been, I have done the same thing for the HDBSCAN. In this case, we have the mean cluster size and the mean sample. The mean cluster size, yes, and okay, and this is optional, okay, so in fact I didn't use it. So these are the parameters that I set for the HDBSCAN, and then I initialized and fit the model. This is how we initialized the HDBSCAN from the scikit-learn library, and how to fit on the scaled parameter or the scale feature space. And then at the end, we can have also the labels. We can calculate the silhouette score, and in this sense, we have the silhouette score that is 0.29. Okay, so it is not so good. We can visualize how the HDBSCAN is performing this clustering, and as you can see, there are 2 points of noise, and all the other points are considered are regrouped in only two clusters. Also, in this case, we can play with the parameters with the mean cluster sample and the mean cluster size and the mean sample, in order to see at the variation of these points how it could change the shape of the clusters, and as you can see, the HDBSCAN is able to separate too many points as noise, and this is the difference with the HDBSCAN. We can try to minimize the minimum cluster points or to maximize them, but nothing is changing. And we can do also, in this case, the evaluation of the optical clustering for the HDBSCAN in order to see. We can see in the first plot with the DBSCAN. I didn't say you that the white cell cells were combination of parameters in which we don't have the solution because we don't have the clusters, so it is impossible to calculate the silhouette score or the Davis-Baldwin index. Okay, so in this case, or in this configuration with the HDBSCAN, we are able to calculate for every combination of the mean cluster size and the mean samples the silhouette score and the Davis-Baldwin. And also in this case, the silhouette is good for blue value around 0.40. I think that these are the best values around, with mean samples around 2 and 6, and the mean cluster size that can vary in this order. And the Davis Baldwin is good for yellow points because they are close to 0. In this case, the minimum is quite around 0.5, and in fact we have the yellow cells that are that is this one 0.5 with mean samples 2 and mean cluster size 2. So also in this case we can use these parameters in order to see how the HDBSCAN is working. And I think that they are these cells with this. Okay, we are seeing here how the solution of the HDBSCAN, in which we are using the best silhouette parameters with minimum cluster size 2 and mean samples equal to 2. And as you can see, we have 89 clusters in this case. So the HDBSCAN fragmentates the clusters, okay, but you can see that all this part is separated. You can see that this part is separated by this one, that there was the part of the parameter space in which the two clusters are nearest. And it is very nice to see also that at the noise point, the red ones, the noise are the red ones, you have the lowest probability of the point to belong to that clusters. If you remember, one of the parameters of the HDBSCAN was the probability of each point to belong or not to a cluster. And as you can see, the noise point obviously have the probability of equal to 0 to belong to a cluster. And it is very useful because, for example, if we want to inspect, for example, here, what is happening to these points that belongs to different clusters, why? And as you can see here, there is a sparse region, the famous sparse region that we were talking at the beginning. And you can see, in fact, that some of these points that are those ones, for example, this one, these clusters with this one, and in correspondence are those ones. You can see that, for example, these 2 points have a low probability of belonging to the cluster. So maybe if we change the minimum cluster size and the mean samples, then those points could not belong to that clusters in which they are now. Okay, so this representation is very useful in order to see what is happening. And the same thing has been done for the best parameters that we searched for the Davis Baldwin, sorry. And also in this case, we can see that there are 85 clusters, and the red ones are noise. And here also there is the probability distribution. Remember that when we use the machine learning in general, not only the clustering and so on, but all the methods, we can also try to do a different set of experiments, not only changing the parameters and seeing how this the solution varies at varying the parameters, but also we can try to use different algorithms because we can exploit the fact that each of these algorithms, for example, if we have to detect anomaly, but tomorrow morning Stefano will explain you more in detail all this part, we can consider different algorithms in order to exploit the different definitions of what an anomaly is for them. And so at the end, you can then check if a point is an outlier for an algorithm or not, or for example, also in this case, if this point, for example, is an outlier for the Risbolding, but also for the silhouette score. And so you can have another combination of probability in which you can say that a point is or not an anomaly, for example, or belongs or not to a cluster. So you can also make this kind of difference. So these are all analyses that we can do with the solution of the different runs of these algorithms. And this is also to answer when this morning we were talking about how is the best choice if we run on some parameter and we obtain these clusters, but then if we vary the values, so then the clustering is changing. And so, well, we can also play with the parameters and understand in this way who is to which cluster, for example, a point belongs or not. Okay, this is the Risbolding, and then also here there is the cluster stability. I measure the persistence, but it is the mean stability. How much stable is a cluster? The famous persistence, and as you can see, stability near 1 means that the cluster is very stable. I colored also the histogram in order to see that, for example, there are the blue clusters that are less stable than the other one. Okay, and this also can be seen for the clustering with the best Davis-Baldwin index, and you can also don't think to the number as the clusters. The numbers the number of the clusters here, the ID, doesn't indicate the correspondence in the plot. Okay, so pay attention to these things. We have always to understand in which cluster there are the points that we want to visualize on the one plot, on the other one. So pay attention to this one, to this thing, and then. Yeah, because for example, I wanted to annotate the number of clusters on this visualization. For example, here there is the cluster 1 with the best silhouette. So I have to go here. I can, okay. For example, this cluster is very stable, and this is this cluster. This cluster, but you can see, for example, the cluster 17 has a lower mean stability. So the 17, I have to see here what is the 17. The 17, it is this cluster. In fact, we have here these points that have a probability, a lower probability to belong to that cluster. So this is also a kind of analysis that we can do. But we can do this kind of analysis when we have also a few points and that can be projected, and we can project them on two axes using, for example, also a PCA. Okay. Okay, this is done using the other library that is that can be installed by do pip install and HDBSCAN. Then we have to import it on the Google Colab. It is already installed, so you don't have to do nothing. Simply import the HDBSCAN here. Otherwise, you have to install it in this way. And then here, for example, if you, how to say, if you train your model, your HDBSCAN, using, for example, a minimum cluster size 15 and mean sample 5, you have to tell that you want the minimum spanning tree, and that at the end you can build it. I have to run the cell. At the end, you can visualize the minimum spanning, the condensed tree and the simple linkage tree, and to see how your data set is splitting in the various or aggregating the same in the clusters. Okay, so you can see how your algorithm is working. But I'm stopping here, Scott, because I can go on and on and on. Now you can, because we are too late, we have 15 minutes, 20, quite 20 minutes to do this exercise. Pay attention to Giuseppe, 18 minutes. There is an exercise that is the unsupervised learning part 3 exercises, in which you can do something. Here there are the libraries. I commented all these libraries in order to help you to understand what you are using and why. But there are 3 exercises. The first one with this kind of distribution of points. The second, and so what is the best methods for example, if you want to understand the methods or and so on. Here there is another one in which you have 3 clusters but a lot of noise, and the third exercise in when you have this kind of structures. So if you want, choose one only to see how to write the code. You can cut and paste the code of my previous notebook. You can do everything. If you want some, if you want an help, we are here. And so only 15 minutes because we have the tour in Naples. If there are not questions. Sì.
Will be fast. You showed us that we can download another package for analyzing deeper the HDBSCAN. That's introduction. And my question is, can we like hack HDBSCAN to select the classes through the alpha parameter directly? Because I see that the alpha, through the alpha parameter, you can understand the hierarchical organization of the classes. So can we hack the method to use this as a free parameter, or like instead of playing with the minimal sample and minimum cluster size, can we also hack and we decide how to define the classes through ourselves cutting the hierarchical tree through the alpha parameter? Yes, that's it.
I think that the best way is always to do a search for parameters, because he is not able. You can use the HDBSCAN or using the default parameters, but there are some parameters that you can leave by default, and you have always to set. And the best choice is to perform a kind of grid search in order to understand what parameters you have to pass him using the matrix, for example, as we have done. Okay.
But we saw that playing with the minimum sample and the minimum cluster size, we saw that we couldn't actually find a good solution to separate the three classes. There was no, well, at least what I play here, there was no way actually.
No,
So
maybe this is the
because
difficult.
the method is powerful. I can say it's powerful,
yeah.
but it seems that if we unconstrain a little bit their parameters, maybe we can make it work better. I
But
don't
these are the methods in which we can constrain.
This is the standard thing. I
yeah.
understand.
I choose to do this problem of clusters that are too near in order to show you that it is quite impossible the research of these parameters in a very
Refined
precise. Yes. Okay.
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 Thank you.
You're welcome.